The captivating realm of cloning has intrigued scientists, ethicists, and the public alike, sparking intense debates and ethical dilemmas. With the power to replicate living organisms or specific genes, cloning promises to revolutionize various industries, from medicine and agriculture to conservation and beyond. However, behind this veil of scientific marvel lies a complex landscape of moral considerations and societal implications that demand our attention and thoughtful exploration. Cloning is a process of creating genetically identical copies of an organism or specific parts of an organism. There are different methods of cloning used in various scientific fields. Here are some common methods for cloning. Reproductive cloning. Reproductive cloning involves creating a genetically identical copy of an entire organism. The most well-known method for reproductive cloning is somatic cell nuclear transfer, SCNT. In this process, at first a somatic cell, any cell in the body except sperm and egg cells, is taken from the donor organism to be cloned, then the nucleus of the somatic cell is extracted. Secondly, an egg cell is taken from another organism of the same species, and the nucleus of the somatic cell is inserted into the enucleated egg cell. The egg cell with the new nucleus is stimulated to divide and develop into an embryo, which is then implanted into a surrogate mother's womb to develop into a cloned organism. Therapeutic cloning. Therapeutic cloning, also known as embryo cloning, is a different process from reproductive cloning. It is not aimed at creating a cloned organism but rather for medical purposes. The steps are similar to reproductive cloning until the embryo stage. Instead of implanting the embryo into a surrogate mother, it is used to harvest pluripotent stem cells for potential medical treatments and research. Gene cloning. Gene cloning is a process of creating multiple copies of a specific gene or DNA sequence. It is commonly used in biotechnology and genetic research. The process involves isolating the target gene or DNA sequence from the source organism's genome, then inserting the target gene into a vector, for example a plasmid, that can replicate inside a host cell, finally introducing the vector into a host organism, usually bacteria or yeast, where it replicates, creating multiple copies of the target gene. Applications. Cloning is widely used in plant propagation. Common methods include cuttings, where a piece of a plant, example, stem, root, leaf, is taken encouraging it to grow into a new, genetically identical plant, and tissue culture, in which plant cells or tissues are isolated, growing them in a nutrient medium to develop into whole plants. It is important to note that while cloning has its applications in scientific research and some industries, reproductive cloning of humans and certain animals raises significant ethical and moral concerns, and many countries have laws prohibiting it. In 1996, the Roslin Institute in Scotland successfully cloned, using the somatic cell nuclear transfer technique, a sheep named Dolly. Dolly's birth was a significant milestone in the field of genetics and reproductive technology because she was the first mammal to be cloned using an adult cell. Prior to Dolly's creation, cloning had been achieved in various species, but only using cells from embryos, not from fully developed individuals. Unfortunately, Dolly's life was cut short. She suffered from health issues at a relatively young age and was euthanized in February 2003 at the age of 6. Some researchers speculated that her early aging and health problems might have been linked to the cloning process or the use of an adult somatic cell. The scientific community is deeply divided over the issue of human cloning, with some advocating for a complete ban due to ethical concerns, while others argue for continued research in non-reproductive cloning for therapeutic applications. As far as human cloning is concerned, it is essential to recognize that ethical considerations are complex and far-reaching. Questions about the sanctity of life, individual autonomy, and the potential for misuse of cloning technologies need to be carefully addressed in any discussions about the future of human cloning. Ultimately, the study of cloning beckons us to ponder fundamental questions about our place in the universe, the sanctity of life, and the impact of our choices on the world. As we move forward, Let us embrace this journey of exploration with a profound reverence for life and an unwavering commitment to making the world a better place for generations to come. The path may be challenging, but it is through understanding, empathy, and responsible science that we will shape a future that celebrates the wonders of life while safeguarding its inherent beauty. By embracing transparent dialogue and stringent regulations, we can harness cloning's power responsibly, preserving our fundamental values while unlocking its benefits for humanity. And that's a wrap on this epic journey. If you had as much fun watching this video as I did making it, don't forget to smash that like button, it really helps the channel. If you haven't already, make sure to subscribe and hit the notification bell so you won't miss any of our future thrilling adventures together. We have so much more excitement, knowledge, and mind-blowing content coming your way. I love hearing from you, so drop a comment below and let me know your favorite part of this video or any cool ideas you have for future content. Your feedback fuels my passion for creating more awesome videos.